Throughout time, organisms have been able to live in inhospitable conditions by random mutations that support the organism in that habitat. One such example is the Venus flytrap. Venus flytraps are said to be carnivorous plants, and this means that Venus flytraps make their own carbohydrates via photosynthesis, but they use carnivory to supply themselves with the nitrogen that they lack through their modified leaves. Venus flytraps usually use photosynthesis in good conditions, but will photosynthesize less when they need to capture their own prey. This is explained by phenotypic plasticity, which is the ability to express different phenotypes when exposed to different environmental cues, in this case, nitrogen. This is an adaptation that came about due to the environment that they live in. Since they live in an environment that lacks certain nutrients in the soil, that they have to obtain those nutrients, specifically sodium and nitrogen, from an outside source. They do this by digesting living organisms such as crickets or flies. But one has to wonder how a Venus flytrap can tell when something is a nutritious source, and if it is, how it can then close to digest it without a digestive system or a central nervous system. The experiment explained in this video studies how the Venus flytrap decides when to invest energy in closing its trap based on the action potentials it receives.
This experiment was conducted using non-invasive surface potential electrodes, with one silver electrode taped to the outer trap and the reference electrode in the soil. Patchmaster software was used to quantify the results. Crickets ranging from 6 to 12 millimeters in length and weighing an average of 23.8 milligrams were used to stimulate the fly traps. Only 2 stimuli were required for the fly trap to activate the closure of the trap, and once closed, the prey continued to trigger the sensors of the fly trap. In order to measure when depolarization occurred within the trap, unstimulated and previously stimulated traps were tested with 9 concentrations of sodium buffers, ranging from 0.3 millimolar to 120 millimolars of sodium. The unstimulated traps depolarized between negative 166 millivolts to negative 99 millivolts, while the previously stimulated plants depolarized between negative 122 millivolts to negative 41 millivolts, displaying much higher sodium dependency. The depolarization of the trap allows the sodium channels present in gland cells to acquire nutrients from the prey, where it is then stored within the plant.
The movement of the sodium ions throughout the Venus flytrap was monitored by placing 5 millimolar of sodium fodder paste on the Venus flytrap's trap. This stimulated the sensory hairs and thus initiated digestion. This was done to see how sodium concentration in the plant changed over time. This is shown in figure 2a. The levels of sodium really began to spike up after approximately 6 hours and increased until 12 to 24 hours, where it reached its maximum concentration of 8,000 parts per million, and remained around this level for 2 to 3 days. This shows that the plant receives its nutrients from the sodium-rich source. The sustained electrical stimulation induced by the prey increases the transcription of jasmonic acid receptors. A 5-fold increase was experienced in stimulated traps compared to non-stimulated.
Within the jasmonic pathway, jasmonates are signaling molecules that regulate diverse processes. To test digestion in this carnivorous plant, the expression of DMHKT1 as a function of induced APs were observed. DMHKT1 is an animal-type sodium channel sequence ortholog that was found in the Venus flytrap. Orthologs are genes in different species that evolved from a common ancestral gene by speciation. The expression of DMHKT1 with no fodder paste but with 3 action potentials applied had no expression of DMHKT1. However, when the stimulus of an insect was applied by creating 50 to 60 action potentials, expression of DMHKT1 increased exponentially. Therefore, sodium is taken up into the glands by the expression of DMHKT1 in the jasmonic pathway. So it can be concluded mechanical stimulus triggers DMHKT1 expression and sodium uptake. The sodium selectivity of DMHKT1 was observed when the sodium concentrated fodder paste was exposed to the readily reacting gland cells, and there was no observable change in the amplitude or the shape of the action potential. So the consumption of sodium-rich foods does not affect the action potential. The stimulus does. When a stimulus is applied to the trigger hair, the mechanical energy is converted into an action potential, which travels through the trap to reach 2 target tissues: motor tissue, which initiates the trap closure and stomach formation, and glands of the endocrine system, which process and digest the trapped insect. When treatment with coranotine O-methyloxime, a ligand derivative introduced to prevent interaction of co-receptors and receptors throughout the pathway, also known as Cor-MO, stomach formation was suppressed, even when 60 stimuli were received. Cor-MO prevented DMHKT1 expression, which concludes the necessity of DMHKT1 expression for sodium uptake by the glands. In conclusion, after a series of trigger hair stimulations, it was found that the JA signaling pathway is activated after the second stimulus, but more than 3 action potentials are required to trigger excretion of enzymes to break down the insect inside. The Flytrap sodium channel DMHKT1 was identified as being responsible for the sodium acquisition, where all of these processes are directly dependent on the number of mechanical stimulations received. These modified leaves help the Venus flytrap in their carnivorous quest for nitrogen that they're not being supplied in their environment.